Here we consider the importance of the geometry, the shape of the Watson-Crick base pairs in ensuring the fidelity of DNA replication, that is, in selecting which nucleotide is brought in based on the sequence of the template. This is question 26 and the end of chapter questions in chapter 1 of the textbook. So it asks, how do size considerations forbid GA base pairs in a double helix? Now, size is not something that we normally think about in terms of ensuring molecular fidelity. In the case of DNA, the more chemical characteristics that determine fidelity are 1, the hydrogen bonds, which are chemically very specific between Watson-Crick base pairs, and 2, the base stacking interactions, which depends on the pattern of positive and negative charges, partial charges in the bases. But what this question highlights is that there's a third parameter underlying fidelity. So let me just write down a heading here that these are the factors that control the fidelity or accuracy of DNA replication. And this third factor is the geometry of the base pairs. And if you just look at these two structures of DNA, one from the side and one from the top, and these are both the most common B form of DNA, you will see that the double helix has a very uniform geometry. That's apparent because no matter where you look at the helix along its length, or if you look from the top, you look at how beautifully symmetric it is in cross-section, this must mean that the shape of the 4 base pairs, that is the shape of AT or TA, depending on whether A is attached to the strand you're looking at or T is attached to the strand you're looking at, your base pair is either AT or TA, or CG or GC is uniform. If it were not uniform, you would see bulges and kinks in the DNA depending on the sequence of the DNA, but you don't. That's because all 4 base pairs look the same. So now if we look at the structure of the Watson-Crick base pairs and also at some mismatches, we see something very interesting. So shown here is on the top in color are the 2 of the Watson-Crick base pairs. So let me just identify these. These are the Watson-Crick base pairs. And the important thing that we realize in looking at this diagram is that the width of the base pair is about the same, approximately 11 angstroms in both base pairs. The other thing that you will see here is that the angle of the glycosidic bond, this is the bond that connects the nucleotide base to the sugar. Let me spell that correctly here. The glycosidic bond is approximately 50 degrees in all 4 instances. So no matter which way the base is slotted in, either TA as drawn here in panel A or AT if it's flipped around, the shape in terms of the angle made by the glycosidic bonds and the distance across the width of the helix is the same for the 2 Watson-Crick base pairs that are shown here. Now, nucleotides need not form base pairs in the Watson-Crick way. They can form what are called non-Watson-Crick base pairs. And 2 such are shown here. One is CA and one is TG. And you will see that the glycosidic bond angles are different from the Watson-Crick base pairs, and the distance for these 2 base pairs is somewhat shorter than the Watson-Crick base pairs, so they wouldn't fit smoothly into the double helix. Now, the reason this becomes so important is that the uniformity of DNA, and here's another representation of the uniformity of DNA, is crucial for the DNA to fit into the DNA copying machine, which is the DNA polymerase. So the DNA polymerase recognizes the regularity of DNA structure. So the DNA polymerase is a protein enzyme, and it has a binding site for DNA, which is discussed in detail in chapter 19. And the DNA can fit in the binding site only if it has the regular shape of the Watson-Crick base pairs. And so schematically, if you consider the DNA polymerase as it's working, it's got a template strand, which is shown in the bottom, which serves as the blueprint for adding new nucleotides. And at each step in the replication process, the correct nucleotide must be selected. but the shape of the binding site is such that only the Watson-Crick base pairs can fit in the active site. So this is the active site, and only the Watson-Crick base pairs have the right shape. Van der Waals repulsions result if an incorrect nucleotide attempts to form a base pair with the template base, and the van der Waals repulsion is so strong that the enzyme will actually not allow that to happen. So that's how the size of the nucleotide determines the ability of the polymerase to include or not an incoming nucleotide in the growing